Like before, there's been a decent amount. Let me make sure. Okay, this is good, right? That's loud. I don't need to make it.
Yeah, no, it's good.
Okay. All right, I'm going to go ahead and get started momentarily. Making sure that everyone has all the packets. All of the packets in the sign-in sheet are located at the front. So what you should have, I'm just going to show you what it looks like to make sure that everybody has each of the packets. We have two separate packets for you tonight. There's one that's labeled General Science. You need one of those. And there's one that's labeled up at the top, Anatomy and Physiology. Okay, so you should have one of each of those. Also, there are evaluation sheets up at the front. Make sure that you've taken one of those as well. And just turn those in at the end of the session. And we also have sign-in sheets, so please make sure that you've signed in. If you would like a copy of the presentation, I can email it to you, so just make sure before you leave that you double-check your email is written clearly enough that I'll be able to understand it. You dotted your I's and crossed your T's, because I hate getting rejected with the emails. But just make sure, double-check on that for me before you leave. This presentation is accessible to you, so we're going to be doing general science and anatomy and physiology this evening. And I'm going to start with the anatomy section because that's a little bit more intensive. It takes a little bit longer of a time to go through. So you'll want to have the packet that says anatomy and physiology. What I'm going to try and do as we go through this evening, you're going to see one half of the screen is on the PowerPoint presentation. The other half of the screen is going to be on the anatomy and physiology packet. I've taken some notes. You may or may not be able to see that very well from the back. I can see if I can scroll this in. My presentation goes in sync with this packet. So for the most part, everything that's covered in the presentation is also in the packet. Just a little bit of info about me. My name is Joyce Danilka, and I work over in the ASC. If you don't know where that is, it's located in the L building on our campus here. It's inside of the library on the 3rd floor. And if you need any additional help studying for this exam, or you want to come in, you have questions as you're going to any of these workshops, we have some of the resource books, the practice tests and stuff like that for you to study from, as well as additional materials if there's any particular topics that you're struggling with. So just so you know that's there for you, my contact information is up there, my IRSC email and my Gmail account. Either one of those is good to email me at if you have any questions, or if I happen to not manage to send you the PowerPoint because maybe it went to the wrong email. So go ahead and take that down. If you don't have that, you can email me either one of those, and I'll be happy to send this presentation to you. So next week, you guys are doing test-taking strategies. I'm going to show you some example questions that are related to the topics we're talking about tonight to give you an idea of the test questions. We'll talk a little bit about some strategies specifically for this section, but next week, that's what you guys will be going through as well. Does anybody have any questions before I get started? Okay. All right, well, with that, let me take this away. Oh, one more thing. Next week, or 2 weeks from now, when we come back for the biology, we have created this handout for you. This handout is a key terms handout. It's separated by the topic for your sciences, so one page is for general science and biology, and the second page, you don't have this yet, but you will have it in a couple weeks, is anatomy and physiology. So essentially, this presentation that I'm giving tonight has gone through and highlighted, to the best of my ability, all of these key terms. But this is a really great study resource for you to make sure that you're touching on all the major topics that come from anatomy and physiology, and it's organized by specific system. So you guys will be getting that in a couple weeks. Just want to point that out to you. All right, so tonight, this talk is divided into a couple different sections. For the general science portion, that's a separate packet. We're only going to be covering what's in bold, so only those 4 topics that are listed there. which is evolution, classification, so that's taxonomy. Any of you have taken biology 2, that's what you talk about in biology 2. Some basic information about how to do research, and then human anatomy and physiology. So that one is actually where we're going to start. And essentially what you have to know are all of the systems that are listed here. We're going to be going through each of these systems extensively tonight, giving you an overview. If you've taken anatomy 1 and 2, you're in a good place for this because essentially it's all of the major topics that are involved in each of these systems. All right? So if you have your packets in front of you, we're starting with the one that says General Anatomy and Physiology. Okay. And the presentation goes in order. So the first system that you're going to notice that's listed number 1 and 2 is the skeletal system and the muscular system. So I've grouped this together and just called it the musculoskeletal system. The figure on the right is going to show you a long bone and place a lot of these different key terms. So everything that you see in bold is coming from a key term. So that's an expectation that you should know what that is and what that means. Essentially, what we're looking at when we talk about the musculoskeletal system is the bones, the connective tissues, and muscles. All right, making sure that you know the structures and the functions for each of these systems. So the main function of the musculoskeletal system is going to be support and protection of the internal organs. And also when we're talking about your skeletal system, movement as well. Inside of the bones, you have a couple different components that they want to make sure that you know and understand. So this is the anatomy of bones. If you've taken anatomy 1, you have already seen this. If you haven't, then make sure that you brush up on this before you go and test. Red marrow essentially is located in the long bones. The long bones, for example, being the femur, where blood cells are made, where your red blood cells are made, inside of spongy bone. Osteocytes is a specific kind of bone cell that's located inside of the hard area, the compact bone. And essentially it's very rich in calcium. Other terms that you have to make sure that you know are ligaments. So what is a ligament? What does it do? Essentially it connects bone to bone. Tendons connect muscle to bone, and cartilage cushions bones at the joints. So as long as you have a good general understanding of that, you can see all of those terms placed in that figure, which is why I really like that figure, to kind of put everything all together in one place. All right. When we look at the skeletal system, you're going to see, if you look in your packets right now, the first 3 pages essentially are just an outline, so it's all text. I highly suggest that these packets that we give to you are very, very concise, very good information, that you read through everything that's in the packet. In addition to what's in the PowerPoint, it kind of gives you a visual to go with what's here. However, if you turn past those 3 pages, you get to the point where you see overviews. And right now, these are also all in order. You can kind of go back and forth as I'm going through the presentation, but you see you have a nice diagram here of the skeletal system. And you also have a diagram of the muscular system. So this is definitely going to be something that you've covered in Anatomy 1. Definitely something on this page you want to have a good understanding of those muscles, the types of muscle tissue and everything that's there. So I just want to point that out to you. And as we go through, you're going to have images in those following pages. So the images are to match up with the text, all right? Essentially, though, when we go back and we look at the skeleton is divided into 2 major parts. So when we look at the skeleton, we have an axial part and we have an appendicular part. The axial portion is going to be just the skull, the vertebrae, the ribs, and the sternum. So when you see this, you can see that on the next slide. I'll show you a picture of that. Appendicular, the way that I always remember that is appendicular and appendages. That would be your arms, your legs, your shoulders, your pelvis, and that's easy to remember. So those are the 2 ways that we divide the skeleton. We have to also understand when we look at the skeletal system, the different kinds of joints, and the function of joints is to connect bones to the skeleton, essentially. We have a couple different kinds of joints that you have to know. Sutures are a specific kind of joint that you see in the bones in the skull. This is an immovable type of joint, so a very specific, unique kind of joint. We also have movable joints, and examples that you're very familiar with are ball and socket, which is going to be in your shoulder and in your hip. And then we have hinge joints. So one example is your knee, another one is your elbow, right? And then you have sliding or gliding joints. So specific examples there are going to be the bones in your wrist, the bones in your feet, your ankle area, and then also the bones that are connecting your vertebrae. All right, so we're looking at the divisions of the skeleton and types of joints. This picture just puts everything all together. In blue, you're going to see the axial skeleton. In red, you're going to see the appendicular skeleton. So just two different divisions. In the other picture, it's green and yellow. So you can get the idea there of how it's divided. And that division just kind of helps you study it and know the system as a whole. For the muscular system, you want to make sure that you're familiar with what's in this slide. So we're showing you 3 different types of muscular tissue, muscular types. So we have cardiac muscle, which is located only in the heart. It's involuntary in contraction. So these are all major things that you want to make sure that you know. The type of muscle, the location, and then what type of contraction it is. Smooth muscle essentially is mainly inside the internal organs of the digestive tract and your blood vessels. This is also involuntary. And then skeletal muscle is attached to the bones by the tendons. It helps to move your body. It's a voluntary contraction. All right, so as long as you have a good understanding of that. Your muscle cells use an enormous amount of ATP to power the reactions and power the motion in your body. So make sure you know what that is. And then lastly, for this system, we have to understand a couple different terms and definitions. So the 2 that are very important are flexion and extension. These essentially are muscles that are going to be found in opposing pairs. So I have 2 examples on this slide. We have the biceps and the triceps in the arm. We also have the hamstring and the quads in the leg. All right, and you can see from this slide, the flexors are going to be your hamstring, the quads are going to be your extensors. An easy way to remember this is that flexion is moving it towards the body, and extension is moving it away from the body. All right, so this is what I was talking about. I have some example questions, and these are meant to help you guys kind of gauge the level of detail. So I'm doing a lot of talking, but this is going to help you decide what level of detail do we have to know. These are example questions. So just take a look at this and see what you think. And it's going to be a tendon, okay? And essentially, the strategy that you're going to use for these is making sure that when you're reading these questions, anything that's part of these questions is an expectation that you would need to know. So you need to know from looking at this question, what is epidermis? What is striated muscle? What is nerve? What is tendon in order to be able to answer this question? Okay, essentially, everything that's part of the connective tissue is included in everything that I just talked about. All right. Number 3 is your digestive system. So basically, this is where we're talking about structure and function. So in one hole and out the other, basically, in a nutshell, but you guys have to know everything that's involved here. So the function is to break down nutrients into smaller soluble molecules that can be absorbed into your bloodstream. You have to know what's in bold. A couple different kinds of types of digestion. So we mechanically digest and we chemically digest. So mechanical digestion is essentially what's happening when you're chewing your food, breaking the food down, increasing the surface area. Things like salivary amylase enzymes are starting to break that food down in your mouth before it even gets to your stomach and intestines. Chemical digestion is what's happening when we're breaking it down into even smaller portions. And the ways that we're doing that, which are also in bold, you're going to notice, are through hydrolysis and through enzymes. So I mentioned one enzyme already. Hydrolysis is just the splitting of water, so you want to make sure that you're familiar with those 2 in this process. when we talk about digestion, we want to understand what alimentary canal is. So you're going to look at that slide and see that everything that's underlined essentially is all part of this system. So from the mouth to the other end, everything that's part of that is part of this system. That specifically, when you see that key term, you're going to associate it with digestion, okay? And essentially what you need to make sure that you understand with digestion is how this movement occurs, what's going on, which phases are mechanical digestion and which phases are chemical digestion, what's the role of your accessory organs. And those are going to be your liver, your gallbladder, and your pancreas in this system. All right. The next phase, after it enters in through the mouth, inside of the stomach, you're going to release gastric juice. So you see that's in bold, also a key term. Gastric juice just means it's hydrochloric acid and a mixture of enzymes, specifically proteases, that are basically exactly what the name says, protein digesting enzymes. And what happens in the stomach is that this breakdown happens from that chemical digestion that's going on here, and it creates something called chyme. Chyme is only partially digested food, but it's more liquefied, so it's easier for it to be absorbed. And essentially what happens is it's passed very small increments into the small intestine at the point when it's ready to be released. All right. And then the liver is going to be an accessory organ in the system. The liver's function in digestion is to produce the bile, and the gallbladder and the bile are going to help to break down the fats. Your pancreas is going to help supply those enzymes that I was talking about to help the chemical digestion happen. Inside of your intestines, though, you have a couple very important features to know, and that's going to be the villi. The villi are things that are lining the small intestine, and they help absorption to take place. So they help to increase the surface area of the intestine, and they also assist with absorption of these products back into your bloodstream and your lymphatic system. So the way that you're getting the nutrients back in after your food has been digested. All right. This slide is showing you the importance of knowing when we're talking about digestion, a lot of this is related to the food that you're eating. So you see we have 3 different types of major macromolecules. We have to know everything that's in this slide. So what is the nutrient? What are the enzymes that are being used in digestion? What are the end products, and where is this located? So carbohydrates are being digested by amylase in the mouth. They're also being digested by sucrase and maltase. All of those are enzymes. It's easy to remember enzymes because they end in ASE. That's just one way to see that if you saw it in a test out of context. Your end product that you get when you break the carbs down is glucose. And essentially, this digestion happens complete in the small intestine, starting in the mouth with amylase and then ending in the small intestine. With your proteins, your enzyme that you're looking at there is protease. And proteins have to be broken down into amino acids. So essentially, you're looking at, on the nutrient, you're looking at your macromolecule, and the end product, we're looking at all your monomers. So those are the single simplest units of each of these. If we consume fats, we have to break those down with lipases, and those are going to get turned into fatty acids and glycerol before they can be absorbed. And the location that this is happening is also in the small intestine. So a lot of action in the small intestine. And I have a video for you guys that kind of puts everything all together and should synthesize this whole system.
After viewing this animation, you will be able to describe the pathway and events that occur during digestion within the digestive system and its supporting glands. Digestion starts in the mouth, also known as the oral cavity, where mastication occurs. This is the process of chewing and mechanically breaking down the food bolus. Chemical digestion starts when the salivary glands release saliva into the oral cavity. Saliva is rich in amylase, an enzyme that breaks down carbohydrates. After the bolus has been broken down adequately, swallowing begins. The food bolus passes through the pharynx and into the esophagus. Peristalsis, which is rhythmic contractions of the muscle layers, pushes the bolus down the esophagus. The bolus passes into the stomach through the lower esophageal sphincter. This sphincter normally remains closed, preventing reflux of the stomach contents. The stomach is divided into anatomical regions: the cardia, fundus, body, and antrum. The muscular outlet of the stomach is called the pylorus. The surface of the lumen is covered in folds called rugae. Peristaltic contractions continue in the stomach, churning and mixing the food bolus with hydrochloric acid and the digestive enzyme pepsin. Neural and hormonal stimulation causes the release of pepsin by the stomach, which starts the chemical digestion of proteins. This helps reduce the bolus to a fluid-like substance called chyme. When the chyme reaches its proper softness, the pylorus opens and the chyme is slowly released into the first portion of the small intestine, called the duodenum. Peristaltic contractions continue in the duodenum. It is within the duodenum that the chyme mixes with pancreatic juice and bile. The process of digestion causes neural and hormonal stimulation of the pancreas to release pancreatic juice. Specialized pancreatic cells produce the pancreatic juice. The juice contains digestive enzymes and bicarbonate. The major digestive enzymes are amylase, which digests carbohydrates, lipase, which digests fats, and protease, which digests proteins. When pancreatic cells release the juice, it flows through tiny pancreatic ducts, which lead to the main pancreatic and accessory pancreatic ducts. Then, the juice is released into the duodenum through the major and minor papillae, where it mixes with the chyme. In order to protect the pancreas, protease is released in an inactive form and is activated once it reaches the duodenum. Bicarbonate within the juice acts to neutralize the acidic chyme, which creates a favorable environment for the digestive enzymes to function. Located above and in front of the pancreas, the liver has many roles, including the maintenance of metabolic homeostasis. The liver maintains this because it receives most of the blood flow via the portal venous system from the small and large intestines. This blood contains nutrients absorbed during digestion. The liver processes these nutrients and vitamins and helps detoxify the blood passing through the portal venous system. Another role of the liver is the production of bile, which is a liquid that contains bile salts and other substances. Bile salts emulsify fat within the small intestine. Liver cells, known as hepatocytes, secrete the constituents of bile into tiny bile canaliculi, which flows into larger hepatic ductules that eventually combine to form the right and left hepatic ducts. Bile then flows into the common hepatic duct and cystic duct, where it enters the gallbladder to be concentrated and stored. During digestion, the gallbladder contracts and pushes the bile through the cystic duct and common bile duct, where they join the pancreatic duct at the hepatopancreatic ampulla. The bile then empties into the duodenum through the major papilla. The bile mixes with the chyme and helps to digest fat. The chyme continues to mix with the digestive enzymes as it moves through the duodenum and into the jejunum, the second portion of the small.
Okay, I'm going to go ahead and stop it there. Essentially, you can see this video sort of summarizes everything that you need to make sure that you understand with these systems. So if you haven't taken A&P2, really emphasize that all of these systems that we're going through, we need a very gross general understanding of what happens in the process of digestion. What happens in all of these things that we're talking about tonight. What are the structures that are involved? What is their function? And what are the major things that are being involved? That video goes into a little bit more detail, but you see a lot of the things that I previously have mentioned are coming up in that video, and that really is a summary of the level of detail that you want to make sure that you know for each of these systems that we're going through. All right, so here's some example questions. Again, when you're looking at these, you want to use a strategy where you're making sure that you understand all of the content that's listed. Multiple choice questions are sometimes harder because you have so much extra information. So remember when I told you elementary canal, you always want to associate that with the digestive system. So as long as you remember that key term, you remember all those components that I had listed, you would be able to handle a question like that because you would be able to eliminate everything else, essentially. It's kind of an obscure term, but making sure as you're going through and studying this that you realize and make that connection, you should be fine. The second question, we're looking at the function of the villi. So if you remember back to what I was talking about, what they do actually in the intestine, you look at this question, you should be able to eliminate a lot from this as well. Their main overall function. All right. And that's going to be to provide a greater surface area for absorption. So associate the villi with absorption. So we're looking at major structures and major functions, and that pattern is going to keep coming back. So to be absorbed by cells, proteins have to be changed into, they have to be changed into their monomers. The simplest unit of a protein is called what? Yeah, amino acids. So we talked about carbs. Your monomers are your single sugars. Amino acids are your single units for proteins. Glycerol and fatty acids are your single units for lipids. All right. The next question is kind of a more difficult question. It's more of an applied question. So you see there's a few different styles in the exam. You have a question here where we have to know what the function of the gallbladder is, essentially. Okay, so this question, if someone's gallbladder were removed, what does the gallbladder do in digestion? It's an accessory organ that helps to digest the fats. So you have to know that in order to be able to answer that question. All right. The next system we're going to go through is respiratory system. And what I have here in this picture, we need to know all of the overall components of this system. You see on the far left, overall, everything that's involved. When we get to the center image, we start to see actually the smallest units, the alveoli and the capillary beds where the gas exchange happens. And this third image, it kind of shows you the connection between the two. But essentially, one thing that I want to highlight is if you look at that image, you see the muscle located at the bottom in sort of a dark red color, the diaphragm. We want to make sure that when you go through the process of respiration that you guys understand what's happening with the diaphragm. So when you inhale, the diaphragm relaxes and it pushes downward as your chest expands. When you exhale, you're going to increase the pressure that's in the chest by exhaling to push the air out. And what's going to happen to the diaphragm, that muscle essentially helps to move and to eliminate the air or to allow the air to come in. When you exhale, the diaphragm is going to get pushed upward. So you have to understand how that all works and the whole process of breathing from the nasal cavity down to the gas exchange that happens at the alveoli and the capillary beds inside your lungs. And I do have a very short video for that. And this kind of summarizes everything in 1 minute. So I want to go through this one with you guys.
Many of us have hundreds of things on our minds at any moment, often struggling to keep track of everything we need to do. But fortunately, there's one important thing we don't have to worry about remembering: breathing. When you breathe, you transport oxygen to the body's cells to keep them working, and clear your system of the carbon dioxide that this work generates. Breathing, in other words, keeps the body alive. So how do we accomplish this crucial and complex task without even thinking about it? The answer lies in our body's respiratory system. Like any machinery, it consists of specialized components and requires a trigger to start functioning. Here, the components are the structures and tissues making up the lungs, as well as the various other respiratory organs connected to them. And to get this machine moving, we need the autonomic nervous system, our brain's unconscious control center for the vital functions. As the body prepares to take in oxygen-rich air, this system sends a signal to the muscles around your lungs, flattening the diaphragm and contracting the intercostal muscles between your ribs to create more space for the lungs to expand. Air then whooshes into your nose and mouth, through your trachea, and into the bronchi that split at the trachea's base, with one entering each lung. Like tree branches, these small tubes divide into thousands of tinier passages, called bronchioles. It's tempting to think of the lungs as huge balloons, but instead of being hollow, they're actually spongy inside, with the bronchioles running throughout the parenchyma tissue. At the end of each bronchiole is a little air sac called an alveolus, wrapped in capillaries full of red blood cells containing special proteins called hemoglobin. The air you've breathed in fills these sacs, causing the lungs to inflate. Here is where the vital exchange occurs. At this point, the capillaries are packed with carbon dioxide, and the air sacs are full of oxygen. But due to the basic process of diffusion, the molecules of each gas want to move to a place where there is a lower concentration of their kind. So as oxygen crosses over to the capillaries, the hemoglobin grabs it up, while the carbon dioxide is unloaded into the lungs. The oxygen-rich hemoglobin is then transported throughout the body via the bloodstream. But what do our lungs do with all that carbon dioxide? Exhale it, of course. The autonomic nervous system kicks in again, causing the diaphragm to ball up and the intercostal muscles to relax, making the chest cavity smaller and forcing the lungs to compress. The carbon dioxide-rich air is expelled, and the cycle begins again. So that's how these spongy organs keep our bodies efficiently supplied with air. Lungs inhale and exhale between 15 and 25 times a minute, which amounts to an incredible 10,000 liters of air each day. That's a lot of work, but don't sweat it. Your lungs and your autonomic nervous system have got it covered.
So from that, you're able to see overall, when you look at the respiratory system, you want to make sure that you understand the movement of air from the nasal cavity through the trachea into the lungs. What happens to that air, that oxygen, once it gets to the lungs, the gas exchange that occurs at the capillaries, making sure that when you look at these systems, we're really looking at the overall big picture. So it's not quite the level of detail that you have seen if you have taken A&P2, but it's just a general overall understanding of the major players and their key functions and how that system all works together to accomplish its end goal. So this, I'm going to show you this website. This slide, I have this link in the presentation. It's a really clear and concise site if you do some extra studying on the side. That kind of goes through and it explains inhalation, it explains exhalation from the National Institute of Health. It also goes through, there's a short animation that's part of this presentation here. So I highly recommend that. It's really helpful to kind of further explain this if you're in a place where you haven't necessarily taken anatomy and you're going to go take this exam. All right, so here's an example question for respiration. Yeah, it'll be, the link should be connected in the presentation. So I'll email you the presentation if I have your email. All right, so in humans, if the diaphragm is pushed upward, there is a decrease in the chest volume. This decrease is followed by, this one's kind of difficult. So go ahead and take a look at those options. Try to identify with the diaphragm which process it's talking about, inhalation or exhalation first, and that kind of helps. Okay, so it'll be increase in the pressure. So you sort of talked about this in the chemistry discussion, pressure and volume, and that kind of relates to that, another example. All right, so I'm going to move on to the next page here. Now we're going on to circulatory system. So when we talk about circulation, we're actually talking about two different systems combined. So when we say circulatory system, we're looking at the cardiovascular system and we're also looking at the lymphatic system. And the function of these systems together is to transport fluid, to help with gas exchange, help nutrient absorption, and to dispose of waste collectively. So we're looking at the overall big picture and the function of this system as a whole. What is this system and what does it do, essentially? And one of the things that you have to be able to understand, if you look at your packet, you have vascular tree. Essentially, 1 and 2 are asking you to understand the movement of blood through the heart. So we have to know that sequence of blood movement through the heart, taking it to the lungs and bringing it back again. That is listed in this slide, but I want to go through this. I have a very short animation that's going to show the whole process essentially, and making sure that you understand to the level of detail essentially that's in this video.
The normal heart has two sides, a right side and a left side, and four chambers, the top receiving chambers, or atrium, and the lower chambers, which are thick-walled pumping chambers, called ventricles. Red blood cell will come from...
So you have to know what the atrium is and what the ventricles are. So he just introduced the atrium in the top. So we want to know the gross anatomy of the heart, all the parts of the heart, and you also want to know their location as well.
Either the superior vena cava or the inferior vena cava, and enter into the right atrium. The blood...
And you're going to notice if you follow along in your packet, I've tried to highlight everything majorly. So we're coming in from the superior inferior vena cava, entering into the right atrium. Right now, what we're talking about is blood that is unoxygenated. So you have a description here, making sure that you understand the location and the movement of blood.
And ejects that blood cell into a vessel called the pulmonary artery. Pulmonary artery splits into two vessels, each going to the lungs. As that red blood cell makes its way through the lung, it returns through the pulmonary veins to the left atrium. That blood is now oxygenated. It's picked up oxygen, then goes across the mitral valve into the left ventricle, which does most of the work in terms of delivery of blood flow to the body. That blood cell is now ejected into the aorta to some organ or muscle or skin in the human body.
Okay, so that does a really good job in summarizing the movement of blood through the heart. So that's one major objective that you have to make sure that you know. If you look through your packet, you can kind of follow along with that. Unoxygenated blood is going to enter the right atrium, coming from either the superior or inferior vena cava. From the right ventricle, it's going to travel out to the pulmonary arteries, where the gas exchange in the lungs happens, that gas exchange that we just looked at happening in the capillaries, right? Then when that gas exchange occurs, the blood has now been oxygenated. It's going to return to the heart by the pulmonary veins and into the left atrium. This portion is going to then proceed on to the left ventricle, and then the oxygenated blood from that point is going to exit through the aorta onto different areas in the cells and different parts of your body. So that general understanding of the movement of blood throughout the heart is something that you want to make sure that you have a good handle on from this section. And much like the other link that I showed you, I have a link for this as well. Same website, it goes through and it explains. I really like this because it's a decent amount of detail, but it's a level of detail that I think is consistent with what you're expected to know at this point. So again, it has an animation, it has a little bit of background information, and it's very helpful to kind of consolidate that if that's something that you haven't taken yet. Okay, another thing that we have to see from this system is when we talk about this, we actually have a couple different types of circulation, and it depends on where the blood is located. So when we talk about coronary circulation, we're talking about circulation of the blood through the heart, and that makes sense if you associate coronary with the heart. Pulmonary circulation is specifically circulation of blood through the lungs, and that's pretty straightforward. And systemic circulation is the circulation of blood through your body. The only other thing to mention here is just the difference in the pressures. Any of you that are currently clinically practicing or working in any kind of medical offices are familiar with blood pressure, so we want to make sure that you know what's the difference between systolic and diastolic, and that all relates to the flow and movement of blood throughout the heart and those contractions that are taking place to actually pump the blood to the different locations in the body. All right, so our major players here, when you look at your packet, arteries, capillaries, unoxygenated blood, we're essentially looking at all of these as components of the circulatory system. So making sure that you know the capillaries are exchanging the gases and the fluids and chemicals in and out of the cells. The unoxygenated blood is being carried by the capillaries and the veins to the vena cava, and understanding how these are actually all connected. So looking at the respiratory system, what's going on there, and how that's impacting the circulatory system. So making sure that you guys understand what veins and arteries are, what a capillary does, and what that network kind of connection showing in this picture is showing you. So we have veins carrying blood toward the heart, and we have arteries carrying blood from the heart, away from the heart. All right, the other component of the circulatory system is in part C at the bottom of your page 2 here. The liquid, the blood system, lymphatic portion. What we're looking at are cells are suspended in plasma. So in your blood cells are going to be suspended in something called plasma. If you look in your packet, when you talk about plasma, it's going to contain the blood cells, but plasma is also going to house a lot of the nutrients, hormones, gases, and waste products that are present. And we want to make sure that you have an understanding of the different types of blood cells. So RBCs, the technical name there is erythrocytes, red blood cells, are making up 45% of this mix. Also within that are your white blood cells and your platelets. Essentially, we want to know what they are and what their function is. So we look at the red blood cells. Mainly, they are the oxygen carriers. The red blood cells have your hemoglobin that helps to carry this oxygen to different areas of your body. Your white blood cells are a very small portion or percentage of your blood overall, but they have an extremely important function in immune defense. So those are going to be comprised of your leukocytes. You have a couple different types, and you'll learn about that more as you go along, but at this point, that's really all that we're looking at. And then your platelets mixed in there are small pieces or fragments of cells. Essentially, they have an important role in blood clotting. Okay, so as long as you know what they are, what they do, and where they're located, those percentages are something that you have just a relative understanding of. You look at the composition and the anatomy of the blood. All right. Within this system, we also have the lymphatic system. So some fluids and proteins are going to be lost as the blood passes through your vessels. And the fluid that leaks out is going to diffuse into your lymph capillaries, and that will then enter your lymphatic system. So this fluid or lymph then can return to the circulatory system. And within that system, you can kind of see, if you look at the image in the bottom in green, you see this lymphatic system, a diagram of the lymphatic system. The larger areas that sort of you can see in the central image are going to be lymph nodes. And lymph nodes are special pockets where the lymph is filtered. So as this is passing through, white blood cells are going to be present inside of the lymph nodes, and they're going to be able to attack any bacteria or viruses that are in that fluid circulating throughout your body. And this is the exact reason why if you're sick, sometimes you end up with swollen lymph nodes in your neck. You can actually feel that. And that's the reason what's going on there is that immune defense reaction happening. So that collectively compromises your circulatory system. So the major components are understanding the flow of blood, understanding what blood is made up of, and then understanding what's going on with that lymphatic system and what it does. Okay? I'm going to be moving on in the packet to number 6, to the excretory system, the renal. Okay? And what we're looking at here, we're essentially talking about the kidneys. The kidneys are the major unit for excretion in the body. So when you look at this figure, you see at the top right, the kidney. When we go down below that, we see the nephron. The nephron essentially is a small component of that. So you have multiple nephrons within the kidney. That is what we're going to call the functional unit of the kidney. And even further, if we take that image, we're looking at, on the far left, bottom left, we're looking at exactly how the filtration is happening. So 1, 2, and 3, the proximal tubule, the loop of Henle, and the distal tubule, all interwoven with those capillaries and those veins, are going to be important in you understanding the sequence of filtration that's happening with the kidneys. So what do we need to know? The kidneys are the principal excretory organ of the body. The nephron is the functional unit. So all those portions that are in bold are key terms that you're going to want to know. So the glomerulus, the proximal convoluted tubule, Bowman's capsule, loop of Henle, and distal convoluted tubule. So when we go back to this picture, number 1, number 2, number 3, Bowman's capsule is essentially where it starts. You can see that in the figure on the bottom left. Okay, so all of these are components of that specific filtration that's going on inside of the kidney. So the kidneys are able to absorb any small soluble particles that are found in high concentrations in the blood. And then essentially what's happening with this system is they're filtering those out. So essentially what is beneficial to the organism is going to be kept. Those waters, nutrients, and ions that we need at this point within the kidney are able to be reabsorbed by your blood. So it's a last check to make sure that you're keeping all the nutrients that you need. And the way that that's happening is by diffusion and osmosis. So if those are things that are foreign to you, you want to make sure that you know what that is. Simple diffusion, areas of high concentration to low concentration. Osmosis, the movement of water to dilute a solute. And then active transport, we have to have energy to get that happening. So all of these processes are happening in order to get filtration happening in the kidneys. The beneficial components are hopefully reabsorbed so that we keep those nutrients. And the things that we don't need are going to be excreted. And this is where we're talking about more key terms. Urine is the concentrated mixture of wastes, so what it is and what it does. Getting rid of those wastes is going to happen by moving that urine to the ureters, to the bladder, and then out through the urethra. So you have a general picture here. You also have another diagram in your packet as well. I mentioned this in the beginning. You have all of these basic figures that are going to go through, and you want to make sure that when you study these packets, you go through and look at everything that's here because it condenses everything into one place. So you see you have that diagram. Understand the movement and how that excretion takes place. Okay, so here's an example question. Most of the work done by the human kidney occurs in the, which one would it be? We have to be able to go through this list and eliminate. Associate, which of these, first of all, are associated with the kidney? Nephron and ureter is all part of the system, but the only one that's actually part of the kidney is nephron. So you see you can eliminate a lot of your potential choices, and that definitely is a strategy here as well. All right. Next system we have is going to be the nervous system. And when we talk about the nervous system, we want to make sure that you understand the two divisions. And so when we divide this up, for our intents and purposes, we're talking about the central nervous system, which is made up of the brain and the spinal cord. And we're talking about the peripheral nervous system, which essentially is everything else. So communicating signals to and from the central nervous system to the rest of the body. In red is the main function of the peripheral nervous system. And these are listed in order in your packets. Function of the central nervous system is essentially to sense, to interpret these signals. You have, if you look in the slide, two subdivisions. It's also in your packet as well. The somatic peripheral nervous system and the autonomic. So the way that I would suggest labeling that is external and internal. So somatic is external and autonomic is internal. That's also in your packet as well on the following page. So you can see all of that listed here. All right, I'm going to go through in a little bit more detail the central nervous system. So when you look at your packet, you see that you have three components that are listed, 1, 2, and 3. I have them highlighted in orange in my packet, so if you're kind of following along with that, you can see it. The cerebrum is going to be the largest portion of the brain. Okay, when we look at this, we're looking at the beige portion that's in the figure, where we have higher level thinking, voluntary actions. We have all kinds of things going on, vision, hearing, speech, memory, controlling all of these things in the cerebrum. The cerebellum is the portion at the back base portion of the brain where muscular coordination and balance. So you want to make sure that you know where it's located and what its function is, what's going on there, understanding that these are all part of the central nervous system. And then the last major component here that we're looking at is the brainstem, located at the bottom. You can see that it's labeled in the green color on the figure. And this essentially has a huge role in homeostasis. And homeostasis, we're going to talk about that a little bit later, but it's just making sure that you're maintaining your body temperature, your blood pressure, making sure that everything that's going on in the body is all regulated and good. The other part of the central nervous system is the spinal cord. And essentially, this is moving from the base of the brain downward. Your vertebrae, your vertebral column, essentially has a hole that houses the spinal cord to protect it from any kind of damage. And it's going to have the ability to pass messages to and from the brain to act at centers to have different reactions or reflexes. And you can see this figure is kind of cool. It shows you at the different regions. It may be a little bit hard to see the small font, but essentially it shows you at the different regions of the spinal cord what different areas of the body are associated with that. So for example, when we get down to the lumbar portion, the lower back area, we see kidney, bladder, and all of those things. So you can kind of get the connection between these two. But the openings that are in between each of the vertebrae are going to allow the nerves and the spinal cord to join together and to create that connection. All right, so we have to understand what is involved in the nervous system. So in this particular system, the neuron is the functional unit of the nervous system. Essentially, you see that is going to be listed down at the bottom of this page. These are nerve cells. These are the nerve cells that are going to be receiving the stimulus from different areas of the body. And they have the ability, they have electric potential because of sodium and potassium ions that are present that are able to cross the cell membrane. So you want to make sure that you understand that connection, that electrical potential that they have. And they also generate signals through something called an impulse. So if you look at the picture, at the very ends, at the far left ends, the extensions that kind of look like they're branching out like roots are the dendrites. And these dendrites are stimulated by something in the environment or in the body, or even by another neuron that they're connected to. And once they're stimulated, they create this moving charge that travels down. You can see by the arrow that I have, the message travels from the dendrites into the cell body, through the axon, and then to the terminal end. So you want to make sure that all those things that I just highlighted, dendrites, cell body, axon, myelin sheath, that you know what those are, where they're located, and what they do. So the myelin sheath that you can see, it looks like little bubbles. Essentially, it insulates and protects the axon from damage. So it serves as a protective organ. When we look at the neurons, so the neurons, remember, are the functional units, the cells within the nervous system. We have a couple different kinds that you have to know. The kinds that we want to make sure that you know are sensory neurons. Essentially, these are responsible for your sense receptors. So when you touch something, it's the message that's being sent to the brain or the spinal cord. The interneurons are kind of easy to remember because they're in between. They're connecting sensory neurons and motor neurons. And motor neurons are really easy to remember because they're essentially the ones that are triggering the movement to happen. So coming from the brain or the spinal cord, going directly to a muscle or a gland in your body, actually controlling some kind of motion. So motor and motion, that one's easy to remember. So make sure that you know what they are and the function for those. And you can kind of see from this figure the connection between the three. So the message coming from number one, from the dendrites, traveling down the axon into the cell body, eventually ending up at the axon terminals of that sensory neuron, connected to an interneuron. That interneuron, the signal is sent down. That one is connected to your motor neuron, and the signal passes from the cell body of the motor neuron, down the axon, all the way through until it gets to the terminal where it actually triggers the motion or some kind of activity for that to start happening. So the connection between these the three types of neurons and then their function is going to be something important for you to kind of see. When we talk about the connection of the neurons, so between 1 and 2 and between 2 and 3, that connection is called a synapse. That's going to be an important word for you to know. So a synapse is the connection of the neurons that are present. And essentially, your neurotransmitters are going to be chemical messages that are sent across the synapses in order to be able to trigger the end movement in step 3. All right. A couple example questions. Spaces between neurons are called synapses. So interneurons are the ones that join two different types of neurons together, but the spaces in between that connection of neurons is called the synapse. So a neuron that transmits impulses from the receptors to the spinal cord. So you want to make sure that you know the locations of these. Which one would this be an example of? From the receptors to the spinal cord, it would be sensory. Sensory then goes to the interneuron. That message then goes on to the motor neuron, which comes from the brain to the muscle or to the glands. All right. At the bottom of page 5, I'm going through the senses. The first one that we're looking at, again, key terms in bold. You don't have that sheet in front of you, but that's definitely a really handy thing to study with. We'll have that for you in a couple weeks. But essentially the major components of the anatomy of the eye. The cornea, the transparent part, the clear part on the outside of the eye that allows the light to come in. The iris is the part that gives our eyes the color, essentially. The pupil is the black portion in the middle. The lens is located directly behind that, and it focuses the light, just like a camera, focusing the light onto the retina. The retina then is at the very back portion, or innermost portion of the eye. It's a layer essentially covering the innermost portion of the eye. And that is very important because the retina has two different types of cells. And these cells are actually what allows you to distinguish between colors, if we're talking about cone cells, or helps you to see at night, if we're talking about rod cells, also black and white. And essentially, when these cells are stimulated, the message is sent to the optic nerve, and that's how you transmit these images. So this kind of gives you, if you look at this image that I have, it shows you the movement of light, making sure that when we're talking about this anatomy, we're looking at the progression, and we're looking at those main things that I highlighted. So you don't have to know everything that's in this figure, but those main portions, the cornea, the pupil, the iris, the light transmission through the lens, the optic nerve at the back, and then the colored image showing you where those rod and cone cells are located inside of your retina. The retina is a little bit hard to see, but if you look at the image, there's an arrow that's directly pointing to where that layer is going to be inside of the cell, or inside of the eye. All right. Very brief on the eye. As long as you know that major anatomy that's listed here, and you know how that's transmitted, you should be good for that. The ear is the next one that I'm going to go through. So when we look at the ear, the main function here is to contribute to hearing and balance. So we have three major divisions that we have to divide the ear into. If you've taken A&P1, you've seen this already. The outer ear is the part that collects the sound and sends it to the tympanic membrane. So the tympanic membrane, most of you probably know that as your eardrum. The middle ear is actually, the middle or inner ear is where all of the fluids are going to be held. And these fluids, the movement of those fluids inside of your ear is going to be what contributes to the sound. So if you notice in bold, the cochlea, most of you who have seen images of the internal ear anatomy, this kind of looks like a snail. If you look at it, it's part of the inner ear where actually these signals are being transmitted. So there's small, tiny hairs that are inside there that send the signals. and send the sensory neurons to the brain that help to contribute to the hearing. Your middle ear is essentially where you have 3 small bones. So you want to make sure that you know those 3 small bones, the malleus, the incus, and the stapes. And the movement of these bones essentially is what helps to contribute to that movement of the fluid inside of the ear. So when you look at it, you can see how it's divided up, outer ear, middle ear, and inner ear. And looking at, especially in the middle ear, you have the 3 different bones, the tympanic membrane or the eardrum, and making sure that you know all of those major parts of the anatomy of the ear and how that sound is transmitted throughout the ear and how that actual process of hearing takes place. It's kind of interesting to point out the eustachian tube connects to the down towards the throat is where your ears will pop. If you've been on an airplane, this is kind of the area where that's happening. So I have a short video that goes through and shows all of that movement.
The ear has 3 parts: the outer ear, or auricle, the middle ear, and the inner ear. The auricle, the visible part of the ear, collects sound waves and directs them to the auditory canal. The auditory, or ear canal, is lined with cilia and ceruminous glands. The earwax from the ceruminous gland and the cilia protect the ear from entry by foreign matter. The thin tympanic membrane separates the outer ear from the middle ear. Sound waves cause it to vibrate. The 3 tiny bones of the middle ear are the hammer, or malleus, anvil, or incus, and stapes, or stirrup. These bones pick up the vibrations from the tympanic membrane and transmit them to the inner ear. The eustachian tube of the middle ear opens into the pharynx and permits air pressure to equalize between the ear and the outside air. The inner ear is maze-like, consisting of bony and membranous structures surrounded by fluid. The semicircular canals are fluid-filled bony loops that help maintain our body's balance. The cochlea is a fluid-filled, snail-shaped structure that houses the organ of Corti, the true organ of hearing. The fluid aids in the transmission of the vibrations. The organ of Corti changes the vibrations into nerve impulses that are picked up by the auditory nerve. Sound waves are collected by the auricle, or outer ear, and sent to the auditory canal, tympanic membrane, ear bones, and then to the cochlea, where the vibrations are changed by the organ of Corti to nerve impulses and transmitted by the auditory or cochlear nerve to the temporal lobe of the brain for interpretation.
Okay, so we're looking at that connection between those 3 major divisions of the ear anatomy and making sure that from those portions that I highlighted, there was one additional thing they were talking about, the organ of Corti, that wasn't necessarily something that's part of your list, but as long as you have a good general understanding of how hearing takes place and those different anatomical parts that make up each of those divisions of the ear, that is a good level of understanding for this portion. The other part here that they didn't really get into too much is what I have down in the bottom. So this is where you kind of get into a little bit of physics with this. Amplitude of sound. So you might have talked about that in the physics talk. But essentially, when we talk about the loudness of a sound, when you look at those 2 images that are directly horizontal up at the top, as the loudness increases, the amplitude increases. And the amplitude is essentially the height of the wave. So you see the picture on the upper left essentially is the loudness increasing, and the picture on the upper right is the loudness decreasing. So very small amplitude there. So the bigger the amplitude, the louder the sound. Another way that we have to look at sound transmission is by pitch. So when we hear things that are higher pitched, what happens is the frequency. So you can see in the bottom right, the frequency of the pitch will increase if it's very high. If it's very low, the frequency is going to decrease. So different kinds of levels of understanding, making sure you know the different parts that are involved, how that hearing takes place, how it connects to the sensory system in the brain, and then how sound is actually perceived, so loudness and pitch. And here's an example question. So the sound is coming in through the ear, hitting the tympanic membrane, and it's causing that vibration to happen more rapidly. What would that be associated with? Higher in pitch. Yep. All right, second to last of the anatomy is the reproductive organism. So what are we looking at here? What's the overall goal of reproduction? Everything in bold, so we have to know the anatomy, internal and external anatomy for male and female. So what the overall objective is, you have this listed in your packet. Sexual reproduction, gametes that are created, gametes in the male going to be sperm. So how does this process happen? You have all of these key terms that you want to make sure that you understand. What's involved in this process? Where is this process taking place? Where is this located? You have a figure that corresponds with this inside of your packet here at the back. But essentially, the overall goal is to produce the gametes. The gametes then will join together through fertilization, so egg and sperm, and then create the zygote. The zygote, making sure that you guys know the difference between haploid, the gametes, and diploid, the zygote. Bless you. What those key terms mean that I have underlined. Same for female. We have to know the overall anatomy of the reproductive organisms. We have to know different hormones that are involved. And for females, the expectation here is understanding the ovulation, understanding menstruation, and understanding pregnancy in the context of what happens as the egg is traveling through the fallopian tubes where it eventually can be fertilized. What is the overall role of the placenta in pregnancy? So to protect the tissue of the embryo and grow together to help to concentrate and connect the nutrients from the mother to the baby. And then the umbilical cord and what's that's function in pregnancy, so connecting the fetus to the placenta. So all of those components that are involved in the female reproductive system and the male reproductive system, how those sex cells are created, and the overall parts of those two systems. And I'll show you, you have, if you go at, I'm not sure exactly what page it is, but everything essentially, there's a good amount of detail that's here in this diagram. I went through and I crossed the ones that are definitely ones that you want to make sure that you know, but make sure that as you're going through this packet, you're looking at everything, because it really does give you a nice overview of each of these different sections, especially if you haven't taken anatomy yet. So what would the answer be to this one? The organ in the human female that develops to nourish the embryo. So what's the role of the embryo? Or what's nourishing the embryo essentially? The placenta. Okay? So when you look at this question, you want to make sure that you're analyzing and realizing that you're able to define what those other components are, and that you understand what their function is and where they're located, whether or not they're placed with the right system. All right, that's it for the reproductive system. The last system is endocrine. The endocrine system, I talked about this word before. It's extremely important. The endocrine system helps to maintain homeostasis. And homeostasis is going to be something that's really important also in the biology discussion. But essentially, it helps to keep everything regulated. So it keeps the body stable by secreting different kinds of hormones, different kinds of things to your endocrine glands or from your endocrine glands. And hormones that are being secreted are essentially defined as the chemicals messengers that help with different kinds of things like growth and metabolism, reproduction. There's a number of different functions that they have in the body. But those hormones are being secreted by the endocrine system to help keep that regulation going, keep your body stabilized. They can either be sent directly to the bloodstream, or they can be sent to receptors on the outside of your cell, or they can be activated by a stimulus. So there's a couple different ways that that hormone secretion can happen. And a couple of the mechanisms that are part of that are called negative feedback and positive feedback. So negative feedback is essentially the way that your body prevents oversecretion. So you don't want to let out too many hormones, otherwise it's going to create an imbalance. You have to be able to regulate this secretion as well. So when a stimulus in your body goes down, the hormone production is also going to decrease. That is what negative feedback is defined as. So an example of that, one that you can remember in your body, is the regulation of your blood sugar. So when you look at this picture, what's going on is if our blood sugar is high, for example, if we just got done eating, we're going to release insulin. Insulin is going to help the body take up sugar, take the cells, take up sugar from the blood so that we can get that nutrition, we can get that energy from the food that we're eating. If we have an excess amount, then the liver will store that sugar in the form of glycogen. So you see that in that green box that I have. Now, on the contrary, if we have a low amount of blood sugar, we're going to release something called glucagon from the liver, and that from the pancreas, and that essentially is going to cause for the liver to release the stored sugar and break down the glycogen. And how this works as negative feedback, that if glycogen is being broken down, no more is being made. So if one thing is happening, the other thing has to stop. The secretion or the stimulus for the other one has to stop if we're doing one thing. So you can kind of see this interconnection. The same goes for if glycogen is being made because we have an excess, then it's not being broken down. So it gives you an overall connection of how the regulation of blood sugar is an example of negative feedback. There's a lot going on with this slide. So I want to point out something to you in your packet. If you go to the figures and diagrams, because this is in order, we've gone through everything, so we're towards the end of those images. And you see something called the Exocrine System. It has a figure up at the top and a list, A through J, with a table there. There's a lot of information in that table. But essentially what I've done is I've extracted some information from that table to show you kind of what it is that you have to know. So we're looking at, from this slide that I have, different types of glands that are part of this system, where they're located, what hormones they're responsible for, and what their function in the body is. So this is the kind of connections that we need to be making when we study this system. So the pituitary gland, located under the brain, for GSH, FSH, and TSH hormones, essentially, you want to identify that as the master gland that's controlling all of the other endocrine glands in the body. The thyroid glands, located in the neck region on the trachea, thyroxine is the hormone there, and it's going to help control iodine. It contains iodine, regulates the metabolism. Parathyroid is behind the thyroid glands, and you can see that that's controlling calcium. Your adrenal gland on your kidneys, and it's easy to remember if you remember adrenal and adrenaline, and you all know what adrenaline does. That one's easy. So fight or flight syndrome, also to regulate water balance, your blood pressure, sort of going through this list and making sure that you understand what the glands are, where they're located, and what their hormones are that they're associated with in their function. Essentially, this table kind of consolidates all the major ones to focus on, as well as what you have in your packet. All right, and this image, I like pictures because it helps to place everything. So you have this diagram, if you look in your packet, it labels all of these different locations out. And sometimes it's easy, if you remember the location, to kind of put the location and the function together, but this kind of just summarizes all of those glands, locations, and just make sure that you understand what their function and what the hormones are that they are responsible for secreting to help the endocrine system maintain that balance. All right, so one example question. The organs control which of the organs? So parathyroids. So you see that table is important. You want to make sure that you know those different types of glands, where they're located, and what they do. all of what we're talking about here, general overview, structures and functions, and if you have a good understanding of that for the anatomy and physiology section, you should be in a good section. You're going to notice if you're looking at your packet, there's one more section there that I'm not going through, but go ahead and just read through that. That's the skin. That's one last section from the anatomy. It's very brief here in the packet, but we know that essentially we have 3 different components of the skin, 3 different major layers. If you look inside your packet at the very end, it's the very last image that you see. You're going to see it's labeled the Integumentary System. So you see, if you look at this, it has the epidermis, the dermis, and the hypodermis, the 3 major layers, and the different sub-layers that are within there. So you want to make sure that you understand that and what's going on with those. In that system, you've seen this before if you've taken A&P 1 as well. That was part of that class. Okay, that's everything for anatomy. Does anybody have any questions on any of that section at all? Okay, so I'm just going to quickly go through the general science packet. So that's the other packet that we've given you. And I have a couple changes, things to modify here. But essentially, we're going to be talking about a few more topics when we get into biology, but this is just kind of a general science overview, so things that are applicable in all your sciences. So scientific method, you probably heard about this in elementary or middle school. Essentially, we have these 5 steps. So we have to know what question it is that we're trying to answer. So usually we call that a hypothesis. What research is. We begin research on the topic, we make a hypothesis, we conduct an experiment, and then we analyze the data in order to draw conclusions. So this setup is how you design your experiments. You have to kind of go through this process to set that up. That's called the scientific method. You have those steps also listed in your packet as well. And from the scientific method, you want to look at how you're designing experiments. So we're trying to overall understand different research methods. And from this, we have variables. Variables are going to be what we call the measurable factors that are going to change during an experiment. So we can have 2 different kinds of variables, and you want to make sure that you know the difference between those 2. I have a picture to kind of help you distinguish between them if you haven't done a lot of research. But an independent variable is something that the experimenter is going to change. So you have control over that. The dependent variable is going to change in response to whatever it is that you're trying to do. So you don't have control over that. When you do an experiment, you want to make sure that you have something that's called a constant. And a constant is something that's not going to change. It has to stay the same throughout the experiment so that you can compare your variables to the constant over time. All right, and here's an example. So when you look at this, you don't have to know what ferulic acid is, or you don't have to know what's going on, but this picture gives you a graph. At the bottom in orange are different concentrations of a chemical. That chemical is, or the compound is ferulic acid. That's the independent variable. That's the one that the person has control over. So you get to decide what concentrations you're going to use. The dependent variable is what happens actually when you do the experiment. So when you combine this, what are the results that you get? The dependent variable you don't have control over. So that's what actually happens when you run the experiment. Another example of an independent variable would be if you're doing an experiment and you had different temperatures. The temperatures are something that you can control, but what happens during the experiment is something that you can't control. That's going to be the dependent variable. All right. So this is kind of a longer question, but it's an example question that relates to how you're designing an experiment. So it kind of gives you an idea of what you would see from this section. So essentially, they're asking you a little bit of a question about experimental design. And if we were to put a control, we have to understand, first of all, what is a control? Control is something that doesn't change. So what would you expect wouldn't change? We're looking at sugars and we're looking at mold, and we're trying to see the relationship between what's going on. So the only one that really makes sense would be water, because you wouldn't expect that anything would happen. So this would be a good control. The relationship between the water and the mold would stay the same. So to answer this question, you have to understand what a control is. All right, lab safety, this is very basic. If you ever work in a research lab or you work in a clinical setting, you have to be familiar with your surroundings. So fire extinguishers. In labs, we have eye washes and shower stations. If you're in a hospital or a medical facility, you have decontamination zones, you have body wash areas. Locations where alarms are, if you're talking about gases that could potentially be damaging, emergency exits. And then most importantly for you to be cognizant of are signage on patient doors. So you have some patients that may have airborne precautions or contact precautions. All of these things are just components to be mindful of when we start talking about emergency procedures and lab safety. And these are some of the things that you might see. So biohazard bags labeled red so that it's clear to indicate that that could be potentially infectious material. A separate container for sharps so that no one gets hurt when they're transporting them. Signage for floors and doors. And PPE, which is called personal protective equipment, so you're all probably familiar with that. Gloves, goggles, face masks, gowns, that kind of thing. All right. In your packet, we're going on to page number 2. Actually, we have a couple different things here. We're starting on page 2. Microscope, and then you have a figure that's on the following page. So this goes back to Bio 1. We have a couple different kinds of microscopes. So distinguishing between those two and what they do. So here we're looking at a compound microscope and an electron microscope. Your electron microscope, if you look at that picture down at the bottom, the amount of detail that you can see with electron microscopes is phenomenal. It's 2 million times compared to the compound light microscope. So the reason why it's like that is because the electron microscope uses beams of electrons instead of light. The compound light microscope just uses light, and the maximum magnification that we see with that is, or resolution that we see, is 2,000 times compared to how much different we can see with the electron microscope. So going back to that class, you want to make sure that you know the different parts of the microscope. And so we have this diagram that's in the packet, and you also have the image on the slide here that goes through and it shows you the different parts. So just have a general understanding of those different parts of the compound microscope and kind of an idea of what they do. And you guys should be in a place where if you've used a microscope at this point, you have a pretty good understanding of what's going on there and what those things do, if you were asked a question about that. All right, so here's a couple different questions. The first one asking about field of view. So just like the zoom on a camera, as the field of view, as the power increases, so as the magnification goes up, the field of view is going to go down. So you're getting closer and closer and closer to what you're trying to look at. So place that in the context of the first question. A little bit of math involved with this one. Okay, so we have the diameter of a cell is half the diameter of the field. The field is 1.6 millimeters, and you would have to know a little bit about the metric system. So you talked about this in Gen Bio 1 as well. We have to be able to convert millimeter into micrometer. So you see what I've done with that little box. Essentially 1.6 millimeters is equal to 1,600 micrometers, so half of that would be 800. So there's a lot involved in that example. And the next one, the diaphragm. Diaphragm controls light. So you see what I'm talking about, where you want to make sure that you go back over the different parts of the microscope and the structures and the functions of each of these major parts that are listed here. All right. The next topic is scientific notation. So when you look at your packets, We do have a listing in there for you. It has all these parts that I'm talking about. It talks a little bit about the functions. So just make sure you go through and review that material that's inside of your packet. The scientific notation, essentially what you have to be able to do with this is convert numbers. So the top image that I'm showing you is a really large number, the speed of light. And rather than writing out the whole sequence of numbers, we would convert this large number into scientific notation. If you look in your packet, at the bottom it says 471. So this gives you the steps in actually how to do scientific notation, but it's very easy. All that you're doing is you're moving your decimal. And you're going to move your decimal to the left if it's a large number, and you're going to move your decimal to the right if it's a small number. So the example that I have up at the top, we're moving our decimal over until it's a number that's greater than 1, but less than 10. So that's the main rule that you want to focus on. So all the way over, I would move this to 2.9 because that's greater than 1, but less than 10. And then all that I'm doing is counting how many places I've moved my decimal. So I'm counting 4, 5, and 8 is 3, 7, 9, and 2 is 6, and then 9 and 9 is 7, 8 places that I've moved in order to get this. And you just write times 10 to the whatever number of places you moved. And the image at the bottom, it shows you how to do this for a really small number. So obviously, it's not convenient to write that number of zeros. So what you would do is just write it in scientific notation, and all that I've done is count the number of zeros, and that 27 that you see as the exponent is actually the number of places that I've had to move the decimal in order to get my number to a place where it's greater than 1, but less than 10. And once you do that, you've basically converted your number into scientific notation. And this is kind of a cool example. And this is actually a galaxy that you can see even with the naked eye from Earth. But if you see, it's actually not that close at all. So another example of how we would do scientific notation on a large number, you see all my zeros and my 4. Essentially, that's 19 places. And I've moved my decimal 19 places over. Because it's a large number, my exponent is positive. And all that I do is write times 10 to the 19, or times 10 to the however many decimal places. So if it's a small number, your exponent is negative. If it's a large number, your exponent is positive. And all that you're doing is just moving your decimal. All right, and you have a couple different examples. If you go on to the next page, you're going to see that there's practice sheets, practice problems. If you need to go back and review this a little bit, or you want to practice and make sure that you're on the right page with that. And the answers are actually at the bottom of that page. So you can go through this packet and get extra help if you need it on that conversion. Most people are pretty good with that. So the tricky part, and this goes back to Biology 1 also, is the metric system. So in your packet, we have given you a metric ruler, and there are actually 3 different rulers on the following page. And this is essentially a system of prefixes. And if you look in this slide that I have at the top, you have kilo. Kilo is very large in comparison to what's at the bottom of that staircase, if you want to use that analogy, is micro, is the smallest one. One thing that I want to point out is that all of these prefixes are units that we're going to add to some kind of a base. So the bases are dependent on what we're actually measuring. For example, if we're weighing something and we want to get a weight on something that's solid, like that block of gold, it's going to be a gram. So we have a kilogram, a hectogram, a decagram. All these different units, these prefixes, are added to our base units. And you can see those 3 base units if you look at your rulers that are on that handout. If we're measuring a distance, or a length, or running a 5K or something like that, that's going to be in meters. All right, so we've heard of a kilometer, like a 5K or a 10K. That's the unit for distance, the base unit for distance. And then if we're measuring a liquid of some kind, the base unit is going to be a liter. So whatever our base unit is, it just depends on what we're measuring. And all that we're doing with the metric system is we're taking these prefixes and we're adding them to our bases. All right? And so what you see at the bottom of this slide is kind of a statement. So it's usually taught where you have like a little statement that helps you to remember the order of how these go. And that's going to be really important. You want to make sure that you see how these are sequenced in that kind of staircase with your base unit being in bold, kind of centralized. So when we look at this figure, the kilo is 3 places away from the base. And the micro is 6 places away from the base. So when we do conversions, we have to realize how that sequence goes. So I suggest, to make this easy, to remember the statement that's down at the bottom with your prefixes in red. That's going to help you see the conversion. And then the method that we use to actually do the conversion, I'm going to go through and do an example with you, just so you have an idea. But essentially, to understand that if we're going from smaller to larger, we're going to be moving our decimal to the left. And if we're going from larger to smaller, we're going to be moving our decimal to the right. So it's all about the direction that you're going, which is why remembering the prefixes is so important. And this kind of gives you, if you look at the jacuzzi image, that's 1 kiloliter of water, and the small tiny dot that's on the dime is actually 1 microliter. So it shows you the huge difference in size that's part of this system. And I want to just do a couple examples with you guys. So if you go on onto the next page, you're going to see it's titled NLN Metric System Conversions. It actually shows you a couple different methods, and you have the steps that are listed in your handout. So if this is something that you haven't done in a while, or you're having trouble with it, make sure you go back through and review that handout. But on the following page, you actually have some examples. And I'm going to just go through and do this with you, give you an idea of a couple different ones that I've picked out. So if you see from the handout, what I essentially have are a few that are highlighted. And the one that I'm going to do is 2.3 milligrams. I want to convert that into micrograms. Okay. The easiest way to do this is, remember, King Henry died. Unusually stands for the base, so I always write that as a star. By drinking chocolate milk. Okay. Usually you're not going all the way down to pico and nano. Okay? So essentially we want to look at how this divides out. So we're starting at milligram. And milli is going to be located right here, because we start kilo, hecto, deca, base, deci, centi. So this is kind of memorization at this point. This one is milli. But one thing that I'll point out that kind of makes it a little bit easier is all of the prefixes that end in I essentially are going to be after the base unit. So when you look at, I'm going to go back to the ladder so that you guys can kind of see this. When you look at that ladder, we're starting at milli, so we're moving from milli to micro. So how many decimal places are we moving, first of all, when you look at the ladder? If you count. Well, from milli, which is right below centi, all the way down to micro, right? So count each step of the ladder, 1, 2, and 3, right? Does everybody see that? Each ladder, each step, essentially, is going to be counted as 1. So we're moving this from milli to micro 3 places, and we're going down. We're moving to the right. So 3 places to the right, you take your decimal at 2.3, and you're going to move 1, 2, 3 places. So essentially what we're doing is we're adding zeros. So this is going to change from 2.3 milligrams is going to be equal to 2,300 micrograms. And if you use the decimal method, it kind of makes it a little bit easier to manage. But in order to do that successfully, you really have to memorize the order that these go in, or be familiar with that order that they go in. So another example I'm going to do is 1.85 liters. Convert that one into milliliters. So liters is kind of a different scenario. Where does liters fall on that ladder? Base, right? Base unit for volume. So we're looking right where it says base unit is where liter is. And we want to go to the milliliter. So how many steps are we away? 3 again. 3 in which direction? To the right, below it. Essentially, if you look at it in the ladder, you're looking at anything below, you're going to consider that to be to the right. Like if you're looking at it horizontally, and anything above, we're going to say to the left. So we're going from 1.85 liters, we're going 3 places to the right. So you just draw your decimal, move it 3 places over, and essentially we're adding 1 0. So it's going to change from 1.85 liters is going to be equal to 1,850 milliliters. All right. And the good thing about this worksheet here is that you have all of these conversion practice problems, and you also have the solutions at the bottom. So you can go through, kind of look at that instruction, combine that with what's going on here in the slide, and get some practice or some review on that if you haven't seen it in a while. All right. Want to go on to the next page. So you've got your sample problems in your Gen Science packet for metric conversions. And then on the following page, we have temperature conversions. So this is kind of going back to Gen Bio 1 as well. Making sure that you know what I have highlighted here. So what's the temperature that water boils? This is like general information that you should know. What's the temperature that water freezes? And you can see that on the page in your packet labeled temperature conversion chart. We have the Fahrenheit scale and the Celsius scale, making sure that you understand how those are related and how you can convert back and forth using that conversion formula. It's pretty straightforward if you were given that information. All right. When you get on to the next page, this information I'm not going to go through extensively, but I do suggest, like I'm saying, that you read through these packets. And one good way to handle this if you're trying to study for the exam is each week we're covering a different topic. So this week we're doing anatomy and physiology and science. Spend this week like reading through these packets, going over this information because it's all fresh. That way each week you're studying kind of a new topic. So I highly suggest reading through this section on the atmosphere. This is going back into some earth science. If you might have seen this not since high school, or you might not have ever seen this, but you need to have a generalized understanding of the compositions of the gases in the atmosphere and a level of understanding of these different layers. Don't get too hung up on it, but make sure that you are reading through it and understanding. Atmospheric pressure, this is kind of an explanation that goes along with that, so explaining to you a little bit about that, what's going on with that system. And then the next section, clouds, a little bit more earth science. So not a strong emphasis, but just make sure that if you don't remember this from when you've been introduced to it, so these different types, that you go back and just read through these pages and kind of refresh on that. When we get into the next page, we see the headings, evolution and Earth's history. So this is going to be important. On the following page, you have a schema that has an outline of all of those. It gives you a real lot of information. I'm going to kind of go through this and consolidate that in just a second. I'm going to start with evolution. And try and focus you guys on the main things that we need to know from that. So this is touching a little bit on biology. But essentially, Charles Darwin is who we associate with discovering the theory that is behind this, and he is the author of On the Origin of the Species, which is a reflection of his work that he did, where he learned that evolved from ancestors and that evolution comes from natural selection. So you see natural selection in bold. We have to know what it is. Natural selection is the traits that are going to promote or enhance an ability of an organism to survive and reproduce. So this idea of favorable traits that allow for survival and reproduction so that you can have multiple generations. And how that was discovered or how that was realized, one way that we can sort of prove evolution is through the fossil record. So this is kind of the how. How do we know what we know? So it's still considered a theory, which a theory in biology is pretty close to being proven, but theory still isn't 100%. So the evidence that we get for evolution comes from something called the fossil record. And the fossil record is things that are found that we're able to date. Specifically now, at this current point in time in history, we can use carbon dating, which is radioactive, so that we can see how old the fossils or the artifacts are, and we can place them on a timeline, like how many years ago was this from? When did this come from? And that collection is what creates the timeline. And so at the bottom of this slide, you see the sequence of migration from these organisms, fish to amphibians, moving to reptiles, and then moving to the land animals, to the birds and mammals. So the fossil record kind of demonstrated that migration over time. And so from evolution, we got a couple key terms. So a lot of these are pretty self-explanatory. Biogeography, biology, the study of life. Geographical distribution of plants and animals. So geography, we look at geographical distribution, plants and animals, different kinds of life forms. So when you put those two together, it's kind of easy to come up with that definition. Comparative anatomy, what are we talking about when we talk about anatomy? We're talking about structures. Structures are going to be comparable in different organisms. So when we compare the anatomy of different animals, that's essentially what we're talking about when we say comparative anatomy. Comparative embryology, essentially we're looking at comparing different embryos, what they look like, how similar or different they are from one species to the next. And then molecular biology is just biology at the molecular level. So we're getting down into the small molecular level with biology. All right, so those are some key terms from there. Evolution today. So this is relevant to you guys, especially when you look at what's on the left-hand side, organisms that are resistant to antibiotics. So what happened here? These two that I have listed, MRSA and C. diff, used to be, or at one point had the ability to be treated, or in some cases are completely resistant to treatment. So that evolution, that resistance, is something that happened over time. So whatever happened with those two, they evolved to have adaptations where they weren't able to be treated, or they're extremely resistant to all the mechanisms that we have to treat them today. Another example is, from an animal perspective, when you look at something that happened recently, actually in Florida, pesticide-resistant insects. So we had mosquitoes that we've been treating with different kinds of pesticides for a long time. Over time, they evolved to be resistant to the pesticides, so they no longer work. And then there were some different methods that they were trying to come up with to fight that. But these are different aspects of evolution, still happens today. All right, the next section, kind of the last section that I have for you, is the Earth's history. So like I said, you have that outline that has very, very small font inside your packet. I don't think that you have to know that level of detail, so I'm going to go through each of these major areas, and what I have in bold is the thing that I would suggest that you would focus on. So when we talk about the Precambrian era, this is the furthest era away. When you look at that in your packet, you essentially can see that that's at the bottom of that figure. And what we want to focus on from that is single-celled organisms. The Paleozoic era is when we started to see plant, fish, reptiles, and amphibians. And Mesozoic era is known as the age of the dinosaurs. Tertiary period is sooner in the chronology, where we start to see large mammals, apes, primitive humans, and it also is when the Ice Age was going on. And then Quaternary period is basically the closest to us, the last 10,000 years, where essentially man evolved to be able to use tools to grow crops, and this is essentially what we would call the beginning of history. Some people call it the beginning of history. So this figure that I have, this little spiral, kind of shows you what's going on there. It's a little bit hard to see, but I have all of those major areas that we just talked about circled or outlined in red to kind of give you an idea, starting from the furthest point coming to the closest point in history to us. So you can see that if you look at the bottom Cenozoic era with man and going all the way back to the beginning of the spiral, you can kind of place that on that timeline. All right, the last topic. You turn to the next page. This is kind of getting us into biology. So when we learned about all these different organisms, all these different species that existed, there had to be a way to organize them. So when we talk about taxonomy, if you've taken Bio 2, you had to look at this a lot, okay? And so taxonomy, the definition for that is how we organize living things. And we group those things by whether or not they have common characteristics. So if you see at the top, I have domain, and at the bottom, I have species. So domain is the most generalized grouping. It has a large grouping of things all together. And then when we get down to the level of the species, this is the most specific. So we have to recognize this order. This is going to be the order that it always falls in. So one way that you can remember this order is if you say King Philip came over for good spaghetti. That's a way that you can remember the order of how this falls. And essentially, how that actually picture that shows what's going on there. If you look at the bottom of this pyramid, you see we have the domain. And then the next step up is the kingdom, all the way up in that order: domain, kingdom, phylum, class, order, family, genus, until you get to the species, which you see is all the way at the top of that taxonomy. So we're going from a really general domain all the way to a very specific species. And when we name an organism, it's done through something that I have there in bold called binomial nomenclature. So you have a genus listed first, and then the species. And that is the most specific level in the taxonomy when we're doing the naming. So of the 3 domains, what you should know is a little bit about each one. So the 3 domains are bacteria, the first one being bacteria. This is all the prokaryotic cells. So you don't know what a prokaryotic cell is. It's a very simple single-celled organism. And it doesn't have an organized nucleus or any organelles. It reproduces asexually, and essentially all of your bacteria will fall within this domain. Your archaea is a separate kind. These are also prokaryotic, but what separates them, what makes them unique, is what's highlighted in bold. These are prokaryotes that have the ability to survive in crazy places with high temperatures, high acidity, high salinity, sulfuric environments, and so they got their own total grouping. And these are essentially more closely related, actually, to eukaryotes than bacteria are. So they're a totally separate domain. The last one is where we fall, is eukaryotic. So we have a huge group here. It's all the plants, animals, protists, and fungi. And what makes this domain different is that we have the nucleus with the cells, with the organelles. Reproduction and replication of the cells is way more complicated, where you have mitosis and meiosis. and essentially multicellular organisms. So we went from looking at single-celled for our other two domains to the beginning of multicellular organisms. So this picture is really nice because it combines everything that's listed on the taxonomy section of your packet. So you're looking at the bottom in the beige, your three major domains, and this shows you kind of a tree diagram that you've seen if you've taken Bio 2, where it breaks down your eukaryotic domain into your kingdoms. So this shows you all the kingdoms that we're looking at. So you want to see the connection between all those different examples. And then essentially this, we want to be able to know those domains, know those kingdoms, and these are your 6 kingdoms that are listed for the eukaryotes. So the domain eukaryota has these 6 domains: bacteria, archaea, protista, plantae, fungi, animalia. And when we go through, we have to be able to categorize each of these. So bacteria are prokaryotes that have unicellular, they can get their food by making it or by consuming. They asexual reproduction. Archaea are also prokaryotes, but what makes them different is their ability to survive in extreme environments. They're heterotroph or autotroph also. Do you guys know what that means, those words, heterotroph and autotroph, from biology? So heterotroph, it basically gets its food. Autotroph, it makes its food, essentially. Protists are mostly eukaryotes, so they fall in the eukaryotic domain. Bacteria fall in the bacteria domain. Archaea fall in the archaea domain. Protists are unicellular, though, and they get their nutrition a couple different ways that you can see there. Plants are also eukaryotes, multicellular. They are autotrophic because they get from photosynthesis. They have the ability to make their food. Fungi are also in the eukaryotic domain, multicellular, and they get their food from outside sources, reproduced by making molds, mushrooms, and yeast. So you see that's in bold. You want to make sure that you know that, how they reproduce as well. From protists, you want to make sure that you know what algae is, what protozoa is, and understand just generally what molds are. And then lastly, the animalia. We have eukaryotes, multicellular also. This group is getting their food from outside, so these are only considered to be heterotrophic. And then reproduction is by mitosis and meiosis. So that kind of covers all of those domains. It's a general introduction into biology, but gives you a basic overview of that general science section. From your packet, if you're going through that, there's one last page, and that last page that's here, actually, it goes more with the chemistry packet, so I have this labeled and a couple changes for you guys to make, actually. I believe you already had the chemistry workshop. So one thing that's here, where it says ideal gas law at the bottom, you want to change that label to combined gas law. The ideal gas law is actually a different equation, so just go ahead and make that change so that you don't get confused when you study that, but you probably already talked about this during chemistry. The rest of this packet we're going to go through for biology and talk about those topics when we do biology. So that essentially is all that I have for a review for you tonight. If you came in late or you missed it, just make sure if you'd like a copy of the presentation, the PowerPoint that I have, if I have your email address, make sure that it's clearly written so I don't get that confused. And also that you turn in the evaluation sheets. That's just if you don't have one, it's at the front. You can just leave that here and make sure that even if you don't want the presentation, if you sign in, please.